Remission is often celebrated as an ending, a stop to a long and painful journey. But for many, it isn't the end. It's a pause, a calm before something darker returns. As of 2022, 50 million people are living with a cancer diagnosis. Yet the shadow of relapse looms larger. For breast cancer, recurrence rate affects every 1 in 10 person, and for colorectal cancer, the percentage increases to 20 to 30%. For glioblastoma, pancreatic cancer, and late-stage ovarian cancer, relapse affects more than 50% of the individuals. These numbers are not just statistics, they are lives. They are lives of people whose moments of relief were stolen too soon. Treatment plans are improving, but with improvement comes longevity, and with longevity comes vulnerability. Current treatment plans beyond remission are still reactive. They require some sort of a symptom or a visible scan, but cancer does not wait. Relapse is unseen, unpredictable, and often unstoppable. But what if this is not the end?
We have spent years fighting what we can see, the tumor, the scan, the symptom. But what if the real battle lies in what we do? What if a vigilant system lived within us, watching, learning, and ready before relapse even begins? Because maybe the answer isn't just to treat disease, but to rethink how we defend life itself, to move from fighting after the fact to protecting before it even begins. Hope has always been what we cling to, fragile, uncertain, and waiting for signs. But what if we didn't have to wait? What if we could re-engineer hope?
In our search for a reliable way to detect cancer relapse, we wanted something more intuitive, a system that could sense biological changes from within without repeated hospital visits or invasive scans.
That idea became TRACER, a synthetic cell-based system that could detect and respond to early signs of tumor recurrence. The TRACER system has a modular design, and each part has a defined role, from sensing a specific signal to deciding if it truly indicates relapse and finally producing a measurable output.
At the core of TRACER lies a synthetic receptor synapse. It's an engineered transmembrane protein that enables a cell to recognize a specific molecular cue, in this case CD147. CD147 is typically present at low levels in healthy tissues and is responsible for tissue remodeling. But in many cancers, it's markedly overexpressed and contributes to tumor proliferation and invasion. By selecting CD147 as the input signal, TRACER focuses on a biomarker that directly reflects relapse-associated biology. In this design, the TET-off system uses a tetracycline-responsive transactivator, or the TTA, which binds to a TET operator promoter and drives gene expression when tetracycline is absent. The presence of the drug prevents binding and effectively switches the expression off. By leveraging this system, TRACER achieves a precise and reversible control. The genes turn on only under specific signaling thresholds and can be silenced when needed. To further refine precision, a tunable inhibitory peptide is co-expressed. It calibrates the system's sensitivity and response range, preventing activation from baseline noise while maintaining strong induction under relapse level signals, practically setting a threshold.
The TRACER cells stay silent under normal conditions, but under high levels of CD147, they get activated and release a reporter protein called G-LUC or Gaucher luciferase, which is shown to have renal clearance. This provides a clear and non-invasive signal for early relapse detection.
Beyond detection, TRACER also carries a therapeutic element, a secreted antibody fragment that targets CD147. This acts as a stopgap for tumor progression.
Each of these modules, the receptor design, the transcription control, and the feedback regulation, integrates into a unified signal response architecture. However, Engineering and biology requires more than just construction. It demands prediction. To anticipate how these genetic layers interact under real biological conditions, our dry lab simulations model circuit kinetics, activation thresholds, and long-term stability. This refines the system before it ever enters a living cell.
Before we could build TRACER in the lab, we had to find out first if our idea could even exist inside a cell. Our goal was very clear. We somehow wanted very high specificity and sensitivity towards cancer cells, and also to prevent somehow the leaky expression of the synapse. The very first model that we built was very ambitious. It was what we called a dual synapse model. Now this model had two receptors arranged in a cascade, each responding to different antigens. Upon modeling, we discovered a tremendous flaw in the reporter expression profile. Even a small leak, just 10% of the basal activation, produced enough TF1 so that the synapse produced was so high and appeared so early that the cells started behaving out of sync, and what we hoped would be a crisp two-step logic turned into a disaster of false positives. As such, the cascade amplified noise instead of filtering it. And that insight shaped everything that followed.
From that point, our goal was clarity. From a dual synapse system, we came down to a single synapse, equipped with a built-in control module, and that control module being an inhibitor peptide. It could fine-tune when our system is supposed to be on and when it's supposed to be not. We observed at low antigen levels, the inhibitor kept the system shut, but at higher levels, the inhibition was overcome and there was a sharp on and off transition. For the first time, our circuit behaved like a real threshold gate. From the simulation, the pattern was clear. Weak promoters barely crossed 15 millimolar concentration, while the strong one easily exceeded 250, shutting everything down. The mid-range between 100 and 200 macromolar was what we were looking for, the point where the circuit behaved precisely and predictably. These cycles taught us that our models are not just predictions, but tools of design, blueprints that guided every experimental choice that we followed.
Once a system felt robust on paper, we turned to structure, how the molecules themselves behaved. We wanted to tag our inhibitory peptide with a 3x flag. Now, it was a question where to tag it. So we performed some structural prediction to model both the versions, the C-terminal and the N-terminal. The C-terminal tag interfered with the peptide's binding site, a subtle clash but enough to compromise function. On the other hand, the N-terminal tag left the binding domain fully exposed and stable. Every step, every model, no matter how flawed they are, they carried us one step closer to a system that can think, respond, and adapt. All that was left to do was to order the constructs.
Once our constructs arrived, it was time to bring TRACER from design to life. We started with transformation, amplifying our circuits and preserving glycerol stocks. We then proceeded to carry out plasmid isolation to confirm that our DNA was ready for expression. And this is where the action happens. We used HEC293 T cells. They were maintained by thawing and serial passaging until they reached ideal confluency for transfection, upon which we transfected our desired plasmids into these cells. Post-transfection, we set up a culture assay with CD19 ligand to validate our ligand-receptor interactions. After incubating the cells, we did a GFP readout assay to confirm our results. Each step, from culture to the first green glow, showed us that the system we built could function inside living cells. And this is where the data unfolded.
The experiments that we just performed validated what our dry lab model had anticipated. The SynNotch plus receptor with CD19 ligand showed the highest signal output set as the 100% reference. Introducing the inhibitory peptide tip modulated the signal as designed, with CMV tip construct showing 62% reduction, CAG tip 57%, and EF1 alpha tip at 43%. We then performed a further titration experiment of EF1 alpha since it showed the minimal variance. While background activity reduced, the overall expression of fluorescence across all the 5 samples was seen to be equivalent. This showed that the signal had shifted from a inhibitor-mediated regime to a receptor-mediated regime. TRACER responds reliably to its input and can be tuned with promoter precision. However, at every step, we wanted to know how our work in the lab connects to the world outside.
That we had been building, experimenting, analyzing and designing, making sense of biology through logic and data. Everything felt structured, precise, almost predictable. But after a while we started wondering, who are they for beyond the lab? And that's when we stepped out of research, out of stimulations, into real lives. The first people that we spoke to were oncologists or cancer doctors who had spent years on the front lines. Listening to them, we realized something that no textbook could ever teach us. Science is as technical as it is emotional. They told us about the days that they could save someone and the days they couldn't. About having to tell a family member that their loved one might not have a lot of time left and still finding the strength to come to clinic the next morning. One of them said, "Hope is the hardest prescription we write," and that's when we realized maybe our work in TRACER would make that prescription just a little easier to fill. Then came the patient interviews. One conversation in particular stayed with us. A family member talking about the last few weeks with late-stage cancer. They talked about just the smallest of things. It wasn't just grief, it was an extra week, an extra smile, a few more memories. And that's when it hit us. We weren't just predicting relapse, we were, in a way, fighting for time. Time for more laughter, more life. Not every moment in human practices was so hard. Like the day we went to an NGO school to teach science to 4th and 5th graders. We spent that afternoon building curiosity from scratch, talking about why the sky is blue, why the plants are green, and how plants breathe, and what makes us who we are. It reminded us that science does not start in research papers, it begins with curiosity and asking why. And maybe that's what we are looking forward to from the next generation, that they never lose that curiosity. A few weeks later, we visited Kendriya Vidyalaya. The students were older, sharper, curious, and ready to dive deep. We spoke about cells, DNA, and about synthetic biology eventually. That idea that we can design life responsibly to solve real problems. A student asked us, "Can we build something like TRACER one day?" We realized this. That's the future right there. Not in our project, but in the questions that they're brave enough to ask. All these experiences, every conversation, every smile, every story shaped how we saw trees. Human practices wasn't just a checklist for us, it was our mirror. It showed us who we were building for and why it mattered so much. And maybe that's what science really is, the connection between data and humanity, between what we know and who we can help.
We are Team BioEngine,
and this is how we are engineering hope.